Who is this? It's Dolly. Right. Okay, where'd Dolly come from?
Dolly came from.
She was cloned. She was not really, didn't really have a mommy or daddy. She was made in the lab. So she was the first successfully cloned living organism. And part of what we're going to learn about in this new chapter, chapter 14, is how cloning works. Okay, so instead we're dealing with cellular reproduction in this chapter, which means we're going to talk again about mitosis and meiosis, which you covered in like grade, was it 8 or grade 9? Not sure. 9? Grade 8? Okay, and I know Mrs. Ramsey would have been pretty thorough with those, so we're going to look at them, but in a little more detail. Vernon.
So you can clone a sheep. So does that mean you can clone a human?
Technically, yes. Ethically, no. Pardon? Yeah, thank you. Okay, we're gonna explore some of the pros and cons of those sorts of things. There's some people who have taken some of their cells and frozen them in the idea, or people that they love, their loved ones. They've frozen some of their cells in the hopes that someday they could reproduce them, right? But they forget, it means that they're going to be babies again, so they're not necessarily going to be the same person, right? So we're going to look at some of those interesting things, interesting questions, and we're going to talk about things like cancer as well in this chapter, because cancer is basically mitosis going out of control. Okay, so it's going to go fairly quick because you've done a lot of this already, so we're going to look at it again, but in a little more detail. So, this was just kind of to get you started with the chapter. So chapter 14 in your book, Mitosis and Meiosis. To understand why we have mitosis and what it's important in, we need to understand what the cell cycle is. So, have you heard of the cell cycle before? Yeah, kind of, some. So basically it's a cycle that every cell goes through in its existence. Okay, some of them will only go through and be stuck in a certain spot for the rest of their existence, but most will go through the whole cycle and keep going through the cycle, and so that's how they will build other cells, reproduce themselves so that the organism will keep living. Okay, so there are two main stages of the cell cycle. So if you look, this thing around the outside, okay, the big arrow is referring to the first stage, which is interphase. So if we were to look at onion cells at the very tips, if we looked at the onion cells, the very tips where growth tends to be happening a lot, you'd see a lot of cells looking like they're doing nothing. Okay, there's no division happening with them. But then you'd see some that are undergoing division. So we're going to look and see some of those cells and what they look like. So this whole thing is a major growth stage, which includes 3 phases. Okay, so interphase, we're going to look in the next slide, is divided into 3 phases. There's this one, G1, which stands for growth 1. Gap 1 is what it's called, but it's a major growth phase. So right after division, okay, it would come right after the cell divides. So it's kind of smaller. It needs to grow in size. It needs to produce other things in order to exist. So it's going to make more proteins and ribosomes, different organelles that it needs in order to live. Okay, then we get into what's called the S stage. So if you read there, what happens during the S stage? DNA synthesis. Okay, so DNA is going to basically be replicated. It's going to be made, a copy of it is made, because pretty soon that cell is going to divide to make another cell. So we'll have 2 cells in the end. Okay? So after the S stage is the G2 stage. Okay? G literally stands for gap, so gap 2 stage, but it is the second major growth stage that the cells will undergo. This growth stage is kind of getting ready for cell division because the M or mitotic phase, this little phase right in here, I'm going to do it in red. This little phase right here is when mitosis, cell division, the division of the nucleus, and cytokinesis, which is division of the rest of the cell, so the cytoplasm and everything, will give us 2 new cells. So we start with 1, mitosis happens, we end up with 2. Okay? So in order to get ready for that, in G2, the cell is going to make extra centrioles, extra proteins that are used for spindle fibers, things like that, in order to be ready. Doubles a lot of its things so it has enough for 2 cells. Okay, so it's kind of getting ready for that cell division to take place. So, any questions about that part? This next slide kind of breaks it down a little bit further. Okay, so we said G1, I need a different color. G1 right here, the first part of interphase is called gap 1. It's the first major growth phase, and the cell is producing lots of things just for it to exist. The second part of interphase we said was the S phase, and you said the DNA is going to replicate. It's being synthesized. DNA synthesis. That's why they say S. The third part of interphase is G2. Okay, that's gap 2, which is another growth phase. It's preparing to divide, so it's making centrioles, spindle fiber proteins, and other things that it needs in order for cell division. So if you think it's probably going to make more organelles, so there's enough for both cells. Okay, so this drawing, this diagram, is kind of representative of how much time they spend in each of the 2 major phases. So if you think of interphase, the big red arrow, and then, I need another color. The mitotic phase, which does the cell spend most of its time in? Interphase or mitosis and cytokinesis? If this diagram shows relative amount of time they spend, is the bigger arrow for interphase or for mitosis? Interphase. So the cells spend most of their time in interphase where they look like nothing's really happening if you were to look at them in a microscope, but there's lots going on inside really. Okay? They'll spend a very short time in mitosis and cytokinesis, so the M phase, and then they'll start it all over again. Cool G. It varies from cell to cell and from different types of organisms. So your skin cells will reproduce very quickly because we're constantly sloughing them off. That's what we wash off each day. Okay, so they're going to be reproducing very quickly. But other kinds of cells don't reproduce as quickly, so they would spend even more time in interphase. Yeah. All right, we're going to play a game. It works. So you want to have that diagram ready and think of what the cell is going to be doing so we can see, and I'm not sure if it's going to show up in the video or not. We might just have to pause it while we play the game. La 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 la. What's it doing? It's going through the cell cycle. Oh, it's going to be slow today, I think. So each time mitosis happens, We go from one parent cell to two daughter cells.
Can you click them all really
Oh, okay. Can you guys see that, or do I need to make it bigger? See it okay?
carefully?
A little bigger? Where's my zoom thing gone? Not on there. Is that better? Okay, so it says all organisms are made up of cells which constantly divide and multiply. Cells divide when they need to replace injured or dead cells or as part of the development of a fertilized egg. 300 million cells are replaced in our body every minute without our even noticing it. Different cells divide differently. Skin cells divide very often. Liver cells divide sometimes. Cells in the bone marrow and cells lining the stomach and intestine continuously grow and divide throughout a lifetime. When a cell dies, it breaks up and leaves an empty space in the tissue. This signals nearby cells to start dividing in order to fill the space. A receptor in the cell's membrane picks up the signal, sends it through to the cell nucleus, and the process begins. Cell division follows a specific pattern called the cell cycle. This cell cycle illustrates the different phases of division. The key molecules which control and coordinate cell division are called CDK and cyclin. Okay, so just a quick review. M stands for mitosis, so mitosis and cytokinesis happens, the cell divides. G1 is part of interphase. What happens during G1? Growth. Okay, it's going to get bigger. It's going to produce some of the things it needs just to survive. S, what happens in S? Right, DNA, we get a copy of it made so that it's ready for dividing a little later. G2 is also a growth. It's the gap 2 stage where more growth happens. Things are being produced to get ready for cell division, so they're going to make centrioles, other proteins, and things like that. All right, so we're going to be faced with a challenge. Welcome to the cell nucleus. You are our new cell division supervisor. A nearby cell has died and we need you. Please come in. Message from outer space. Cell death in tissue. Cell division signal has entered the cell. New cells required immediately. Okay, what's going to happen? Cell division. New cells required immediately. Wrong choice. Please choose another activity or the cell will be destroyed in 10 seconds. What happens first? The cell has entered the gap 1 phase in the cell cycle. Cell growth has started. Cell at highest energy level. Okay, so it's getting bigger. It's increased by 20%. So these guys are checking. Is the cell size okay? Doop, doop. I don't think it is. It's going to check and see. It's big enough to proceed. Now we got to check the genetic material. Genetic material is checked. There's no damage found. The checkpoint is cleared, so we can proceed. What happens after growth? What's going to happen next? Oop. Chromosome duplication. Cell is entering the synthesis phase. Duplication of genetic material has started. You see, now you have 2 copies of each. The cell is ready to proceed in the cell cycle. What's going to happen next? Cell growth. Cell is entering the gap 2 phase. Continued cell growth. Okay, cell has reached checkpoint 2. So this time we need to check. All genetic material has been duplicated. Has it been duplicated? All chromosomes are duplicated. Please proceed. What happens next? We check the cell size. Cell is big enough to enter the next phase. Please proceed. We're going to check and make sure, is all the genetic material intact? Is it all okay? Does it look okay?
No.
Uh-oh, there's a white thing. Something's wrong. So we need to repair it. Okay, checkpoints cleared. Ready to proceed in the cell cycle. So, cell division, chromosome separation. What do you think? Okay, so this is the mitosis part of the M phase. So the nucleus, the chromosomes are going to be pulled apart, and new nuclei are going to be formed. So in order to pull them apart, we check to make sure that the chromosomes are attached to these stick things called spindles, spindle fibers. Spindles are okay, they're all attached to the right spot. The checkpoint's cleared, so oh, he's happy. Well done.
What would have happened if it's been self-destruction?
Cell destruction? It would have destroyed itself. So we'd have to start all over again. Yep, okay. So cell cycle is strictly controlled so the daughter cells produce their exact copies of the mother cell. It's important that the different phases of the cell cycle are precisely coordinated and that phases follow in correct order. Coordination errors may lead to changes in the chromosome. In other words, you can end up with mutations. Chromosomes or parts of the chromosomes may be lost, rearranged, or redistributed unequally between the two daughter cells, and that can affect how those cells and tissues work. And if it's during development of an organism, like when they're becoming an organism, then it can cause them to have serious consequences in that. In order to ensure that only faultless cells divide, there are special supervisors. We already said they're called CDK and cyclin, which make sure everything proceeds in the right order. If something is wrong, they pull the brake and repair the damages before a cell can proceed. So are they little men running around in you? No. They're chemicals that kind of trigger different things, enzymes and stuff to go and proofread your DNA, things like that. Pardon? If the supervisors are not working properly, so if they're like down and out, not working as they should, then the cells may become cancer cells, which often have altered chromosomes. They may divide uncontrollably and much too fast. These mutated cells finally form a clump called a tumor. Understanding the control mechanism of cell division or cell cycle is necessary for understanding how cancer arises. For one example. Are you worried about the control of cell division in your body? Do you think about it? No. The fact is, it's very rare that anything negative happens, and the supervisors do a good job. So, that is the cell cycle. So we're going to talk more about mitosis today. We'll talk about meiosis another day. Yep.
How do they tell the difference between a benign tumor and a cancerous tumor?
If they look at different features of the cell, they'll usually take a biopsy of it if they can, and then they can look at different features of it under the microscope.
So like a benign tumor, does it not have like the messed up chromosome? Or is that just cancer?
They'll also check and see if the tumor has spread. If it has spread, then it would be definitely a malignant one that has spread to other areas. So they can look at different features of the cells that way, and they'll also check and see if there are any other tumors close by. They'll usually check your lymph nodes, things like that. So, yep. Okay, so Mitosis. So we start with how many cells? 1, and we end up with how many cells? 2. Are they same or different from the original cell? Same. So if you are talking about a skin cell, then that skin cell, as it's getting close to being done, it's going to undergo cell division, and it'll make 2 new cells. Okay? Or if there's some close by that are damaged, then the one close by will divide in order to repair that area. Okay? And they happen in the skin very frequently because they're constantly being sloughed off, and the next layer will be sloughed off the next day, kind of thing. Okay, so mitosis refers to division of the nucleus, so you should know what it means. It occurs in most somatic cells. Okay, so you're going to need to know what somatic cells are. You know what somatic cells are? Your liver cells, your skin cells, your anything that's not going to be able to make a baby. So your non-reproductive cells are somatic cells. Okay, so the results, we start with 1 cell, and we end up with 2 daughter cells that are identical to the parent cell. Why does it happen? You should understand why it's happening. So basically, it can happen for growth, repairing damaged cells, replacement of dying cells, specialization. So when your cells are first forming in you, then they'll start to form and become different shapes so that they can do different jobs, okay, depending on what kind of tissue that they're made in. And it's a means of reproduction. Okay? So in something like when you take a plant and you rip off a piece and then you put it somewhere and you let it grow into another copy of that plant, that is not involving any reproductive cells, right? Don't have any pollen or eggs, ovules for reproduction in that type. They just keep on undergoing mitosis and form more of the plant in order to grow the rest of the plant back again. Yep.
Since human body is meant to regenerate, how can we die of old age?
Since it's meant to regenerate,
Yeah.
we can't regenerate to that extent. We can only repair like small localized little regions, right?
Why is human body die of old age?
Because if we're going to talk about aging and how your cells seem to have little counters of how many times they divide.
Oh.
Yeah. Good question. Okay, so last year we looked at budding in sponges. That's another form of asexual reproduction. So if it's asexual, it means it doesn't involve any sperm and egg or sexual spores. Okay, so it means that it's just a piece of the parent plant or an original cell from the parent plant that keeps on dividing by mitosis to make the new baby or the new plant. And if we are undergoing sexual reproduction, then that gets us to have 1 cell. We all started as 1 cell, right? Okay, the egg and the sperm, they come together and they form 1 cell. But to get that 1 cell to go to the billions of cells that make us up, we undergo mitosis. Okay, so for asexual reproduction, you need both meiosis to get the egg and sperm and mitosis in order to form the organism. These are the 4 phases. Do you recognize those? A little bit? Okay, so we're going to look at them each in a little more detail. Meiosis has the same names for the phases, except it undergoes it twice. So it makes it a little bit different.
Are they in order?
Yep. Did I print this slide for you? Yeah? Okay. Just checking. Okay, so the cell cycle. So features of interphase. So these are just highlighting features of the different phases. Okay, so you should be able to recognize if you were given a cell and you look at it, you should be able to tell what phase that cell is in. Okay, based on different features about it. Or if you're given a description, then you should be able to tell what phase it's in. Okay, so interphase looks like nothing's really happening, but there's lots going on inside. So we said these 3 stages are happening. So gap 1, lots of growth happening. Okay, it's going to duplicate organelles, it's going to get bigger, going to make lots of proteins. Synthesis is when it makes a copy of DNA. Okay, so it makes a copy of the chromosome. And then in the last stage, gap 2, G2 is the last bit of growth and producing of things needed for cell division. In this phase, you cannot see the DNA. Okay, it is super, super coiled up. Okay, it's really, really tiny. We call it chromatin. So the chromosomes, whenever the cell is in interphase, you won't be able to see them. This is kind of showing them, but just to the effect that they're like little strings of yarn, but like not even microscopic. Can't even see it in the microscope. Okay? And the cell looks inactive. So, everybody remember cell parts? I know you guys haven't seen them in a while for the grade 11s. Basic cell parts. So in an animal cell, this outer part is, the cell membrane. These are found in animal cells and not plant cells. They're centrioles. They help to direct cell division. So that's why they're being shown here. They're really important for cell division. Okay, so they would have made an extra copy of them during the gap 2 stage to get ready for cell division. The dark body that has the DNA inside is the nucleus. Okay, so the nucleus is visible, and the nucleolus is usually a dark body inside of the nucleus. Okay, so you wouldn't be able to see the centrioles, but you could see the nucleus. You can see the cell membrane, and it'll look fairly clear on the inside where the cytoplasm is. Prophase. So prophase is kind of the very first stage of mitosis. Okay? So things that happen, the membrane starts to dissolve. The chromosomes become visible. Okay? And we call them in this stage sister chromatids. I'm going to trip myself on this thing. Okay. So this would be one copy of that chromosome. But during the S phase in interphase, what happened? Right, it copies itself. So here is the second copy of the same chromosome, and they're held together by a centromere. Sometimes they call them a kinetochore. Okay, so they're held together so that they can travel together and line up together as they should. So each of these, each one is a sister. So they're sister chromatids. When they're visible, they're called chromatids. When they're not, so during interphase, the chromosomes are referred to as, remember? Not chromatids, but chromatin. Yeah, or chromatin. All right, so what else do we see? The centrioles are starting to migrate to the poles because they're going to be on opposite ends of the cell because they have to pull things to opposite sides. Okay, what are these things, do you think, that are forming? The spindle fibers, right? The spindle fibers are forming so you can see them because they are needed in order to pull the chromosomes to each end. Okay, and this was just showing that, this is starting to disappear. It's breaking apart. Okay, any questions about prophase? Okay, metaphase. Meta means middle. So this one is probably the easiest one to pick out because where do the chromosomes go? In the middle. The sister chromatids line up at the middle. Okay, so if you notice, the centrioles have reached opposite ends. They're at opposite poles. Ready? And the spindle is complete. The chromatids line up at the equator in the middle. And the spindle fibers attached to each one. Where are they attaching? They attach to this thing that's holding them together. What are they? The kinetochore or centromere. Okay? So the spindle fibers are going to pull that apart, and they're going to pull one chromatid in each direction. Okay, so this one would go this way, and then its sister would go the other way in order to make sure that each new cell is going to have one copy of each of the chromosomes. Okay, so the easiest way to pick out metaphase is look for when they're lined up right in the middle. Anaphase. Okay, the centrioles are still at opposite poles. The spindle fibers are starting to shorten because as they shorten, they pull the chromosomes towards the opposite poles. Okay, so the spindle fibers are pulling them. The chromatids look U-shaped. So if you think about if you were swimming in a pool and you had a rope and you were holding the rope in the middle and you were swimming along with it, how's it going to look? It's going to be U-shaped, right? Because of it's pulling it through water. Well, these are doing the same thing. They're pulling them through the watery cytoplasm. Okay, so it makes them form like a U shape. So you can tell that they're moving to the opposite poles. Any questions about that one? Okay, telophase. Telo means end. Okay, so it's the last, technically the last phase of mitosis for division of the nucleus. So if you look, where are the chromatids? They're at the opposite poles. They're in the new cells, basically, the ones that are forming. Okay, they're not completely formed yet. They're at opposite poles, and what happens to the chromatids? Right. Okay, so they're not going to be as visible anymore. They're going to become all tiny little strings. Okay, string-like structure so that you can't see them. The nucleus begins to form, and the spindle fibers will break down. At the very end, we're going to see that a cleavage furrow starts to form. It starts to pinch in on itself. Okay? Some books will consider cytokinesis as being a phase of mitosis. Others say it's a thing on its own. Basically, they happen pretty much together. So cytokinesis is how it's pinching apart so that it divides the cytoplasm. The organelles are divided between the cells randomly. Okay? The cell's not going to take the time to make sure, oh, well, you have 200 of this protein and you have 50 of this. It's not going to make sure it's exactly even. It will end up being that they have enough of each type of organelle, but they're not necessarily going to be 50-50. Okay? So in the end, this would pinch off, and you have new cells that look like the very first cell. Looks like nothing much is happening. Can you guys see this diagram on your slide? Yeah. Okay, so in mitosis, we start with the one parent cell. How many chromosomes are there? 4. Okay, their DNA is duplicated during interphase, and then during mitosis, we get 2 new cells that have how many chromosomes? 4.
The cell cycle is a complex series of continuous events consisting of interphase and mitosis. During interphase, the nucleus and the darker staining nucleolus can be clearly seen. The DNA is copied. The chromosomes cannot yet be seen because they are still in the form of uncoiled chromatin. In animal cells, the centrioles duplicate themselves. In prophase, the chromatin coils to form visible chromosomes. The nuclear envelope and nucleolus disappear, and a spindle forms between the pairs of centrioles, which have moved to opposite ends of the cell. In metaphase, the chromosomes move to the equator of the spindle, with each chromatid attached to a separate spindle fiber by its centromere. During anaphase, the centromeres split, and the sister chromatids are pulled apart to opposite poles of the cell. Each chromatid is now a separate chromosome. Telophase is the final phase of mitosis. In telophase, 2 daughter cells are formed, with a complete set of chromosomes at each end of the cell. The cytoplasm divides, the nucleolus and nuclear envelope reappear, and the chromosomes begin to uncoil. When the new cells are separated, they enter interphase, and a new cycle begins.
Mitosis is a process of nuclear division in which the duplicated chromosomes separate to form 2 genetically identical daughter nuclei. During prophase, the nuclear membrane disintegrates and the nucleolus disappears. As prophase continues, the chromosomes condense and begin to appear. Mitotic spindles, which are made of microtubules, begin to form between the poles, and kinetochores begin to mature and attach to the spindles. During interphase, the chromosomes become attached to the mitotic spindles and the kinetochores and align along the metaphase plate at the equator of the cell. During anaphase, the kinetochore microtubules shorten, separating the chromosomes to opposite poles, while the polar microtubules and the cell elongate. During telophase, chromosomes reach the poles of the cell and begin to disappear. The polar microtubules continue to elongate. The nuclear membranes form, the nucleoli appear, and the chromosomes decondense. Telophase is followed by cytokinesis, during which a cleavage furrow forms in the center of the cell, and the cell divides.
I want you to remember this last little part for when we talk about plants and animals. So,
Nucleoli appear, and the chromosomes decondense. Telophase is followed by cytokinesis, during which a cleavage furrow forms in the center of the cell, and the cell divides.
so what kind of cells are these? They're plant cells. Yep. How can you tell they're plant cells? They have a cell wall, which gives them a rigid shape. Okay, which is usually, it might be rectangular, it might be hexagonal, but usually it gives them a very regular shape. Okay, and because of that cell wall, they have a bit of a difference whenever they go to, they can't just have a furrow form, they have a different happening whenever they go to have cytokinesis. So, This is from your book. Okay, so what phase is this? Interphase. Okay, you can see the nucleus. You can see kind of they're showing the uncoiled DNA. Okay, in chromatin form. You can see the centrioles. They're going to be duplicated, and then here you can see them migrating to opposite poles. Where did the nucleus go? Disappeared. So this is prophase. Metaphase, they're going to line up in the middle. Anaphase, they're being pulled apart. And telophase, they're at opposite ends. The nucleus reforms. Okay, these are going to start to disappear again. And what's this again? The furrow. These are showing the plant cells doing the same thing. If we look, this is this one was labeled with a radioactive dye so that it could fluoresce, and you could see it undergoing mitosis. It's I think it's a fish blastula, so it's in its early embryonic development, so it's undergoing it a whole lot to make a fish. This is the same sort of thing in nerve cells, I think. No, not nerve cells. Pardon? Sometimes they make these faster. It depends on the kind of cell. Skin cells? I think it's once every 24 hours, but I'm not sure. So make sure you bring your headphones for tomorrow. This one was to highlight the differences between plant and animal cell mitosis. So if you look, one of the big differences is animal cells have these, the centrioles, whereas plant cells don't. Okay? The other difference, of course, if you look at them, the shape of the cells themselves, but that's just to do with the cells, not the mitosis. If you look at what's happening for cytokinesis, okay, in an animal cell, the cell membrane will pinch inward. That's called formation of a cleavage furrow. That can't happen because of the cell wall in plants. So instead, they form what's called a cell plate that forms along the middle and forms the cell walls for each new cell. So we reveal there are the differences. Okay, so there's an animation of that. Okay, so this one shows animal cell division, cytokinesis, where the furrow happened. This one over here is showing the formation of the cell plate, which will form two new cell walls. Okay, and those are linked from the website if you want to see them anytime.